Hey everybody, it's me, Mr. Lang again, and today I want to talk to you about microRNA. Yep, it's just another form of gene control. So, you've been through all sorts of different ways that genes can be controlled. And you know that there's all kinds of stuff that has to happen in order for a gene to be transcribed and translated into a protein. You need to have a promoter, you need to have a TATA box, you need to have transcription factors, you need to have DNA bending proteins. There's all sorts of different things that you need. But here's the question. Is it possible to have everything in place that needs to be there and still not make a gene? Well, the answer is yes. So it's basically, I've oversimplified things as we've gone along because it's starting to get more and more complicated. And at this point, it's time to start exploring some of those complicated corners of transcription and translation. So before we do that, I want to spend just a couple of minutes talking about transcription as you know it. So we've got our double-stranded DNA here. We've got RNA polymerase. It's going to come in. It's going to identify a sequence that it's going to copy. That's going to be the RNA transcript. It's going to actually make that copy. We're going to have RNA processing come in. RNA processing is going to cut out sections and put the exons back together to make our finished mRNA. That mRNA is then going to leave the nucleus, my fancy blue line here, that's the nucleus. It's going to attach to a ribosome. The ribosome is going to run along it. It's going to create our finished polypeptide or protein. That's what we've talked about. What if something different happened? What if I identify with my RNA polymerase a sequence of DNA that's further down the same chromosome that we're looking at? That's what this little slash is for. And I copy that, I transcribe it. But because of where it is, there are complementary bases across from each other. So if there's an A here, there's a U here. If there's a C here, there's a G here. If there's a G here, there's a C here. If there's a U here, there's an A here. You get the idea. What happens is you get this piece of mRNA that looks like a hairpin almost. That piece of mRNA then can leave the nucleus just like this. And there's a dicer protein that's going to come in and cut off that curved end, and I'm going to be left with a piece of DNA that looks like this. And actually, it's going to be of RNA, sorry, that's going to be double-stranded, which means I've lied to you yet again, because up until this point, I've told you that RNA is always single-stranded, but in this case, this microRNA starts out as a double-stranded piece of nucleic acid. So here's how this can work. This is the gene control aspect of it. So we're going to go through the process at the same time. We're going to have two different RNA polymerase molecules coming in, one making an mRNA transcript and one making a microRNA transcript. These are two different places on a chromosome. Could be different chromosomes. We're going to go through RNA processing with our normal mRNA transcript to make finished mRNA that's going to then leave the nucleus. After we've left the nucleus, the dicer protein is going to come in and it's going to cut off that hairpin end of the microRNA. And here's where the magic happens. Usually the ribosome is going to come in and start making the protein. Another protein is going to come in and it's going to take off half of that double-stranded microRNA. It should have been double-stranded. I couldn't figure out how to do it. So it's going to take off half of that double-stranded mRNA, and then that double-stranded mRNA with the protein attached is going to attach to the finished mRNA transcript. So This is sort of like a repressor protein on an operon that you've been talking about. Um, with this here, we can't translate this piece of mRNA completely. So this would be a way that I could stop the ribosome from moving along a piece of mRNA, and that is gene control. So I can have all of the stuff that I'm supposed to have in place. I can have the promoter, I can have the TATA box, I can have all of the different aspects that I need to have expression of a gene happen, and I can still stop it if I have a microRNA single-stranded piece in this binding protein that can bind to the transcript and stop it from being transcribed if for some reason I don't want it to be transcribed. So that's how microRNA works. It works in conjunction with a finished piece of mRNA, and it literally, physically, along with this binding protein, stops the ribosome from moving down the finished mRNA transcript. Hope that helps you understand microRNA. Let me know if you have any questions. You can contact me during office hours or in class. Take care.